Welcome to part 1 of chapter 16 on epidemiology. In this section, we're going to build the foundational vocabulary that epidemiologists use to describe disease in populations. This chapter has 4 sections. Today, we're focusing only on 16.1, the language of epidemiologists. We'll define key terms like incidence, prevalence, morbidity, mortality, and the different patterns of disease occurrence. By the end of this lecture, you should be able to 1. Distinguish between prevalence and incidence, 2. Describe sporadic, endemic, epidemic, and pandemic diseases, 3. Explain Koch's postulates, and 4. Connect epidemiology to public health systems. Epidemiology studies who gets sick, where, when, and why. The goal is to recognize patterns, identify the etiology or cause of disease, and control outbreaks. We're moving beyond individual patients and now looking at disease behavior in entire populations. Epidemiologists describe diseases within a population, and this could be a geographic area, an age group, or people sharing a behavior like smoking or an occupation like all of the nurses in one city. All disease numbers must be interpreted in the context of the population at risk. A raw number of cases is meaningless unless we express it as a rate because populations will differ in size. For instance, if we have 500 cases of a disease in a small town with only a few thousand people, that would be a major outbreak. But if we had 500 cases in the entire United States, which has over 340 million people, that would be a pretty low rate, pretty low occurrence. Now let's get into some of the vocabulary related to epidemiology. First, we have morbidity. Morbidity simply means having the disease. Total morbidity would be the number of cases, but without population context. Morbidity rate is better because it shows how often the disease occurs in a given population. And these rates are standardized, usually per 100,000 people, so different regions and time periods can be compared meaningfully. Morbidity rate also reflects the disease burden in a population, and this could have economic consequences if people cannot work consistently, if they can't maintain employment, and they can't support a growing economy if they're constantly sick. Our next vocab terms are prevalence versus incidence. Prevalence counts all existing cases at a point in time. For example, the book states that there were about 1.2 million HIV cases in 2012, or when expressed as a rate, 467 per 100,000 individuals. More recently, about 1.13 million total people in the U.S. were living with HIV in 2023, according to the CDC. Incidence, in contrast, refers to new cases only within a set time period. For instance, here the book provides a number around 43,000 new HIV cases in 2012, or as a rate, 13.7 per 100,000 people. More recent numbers here indicate around 39,000 new HIV cases in 2023, but if you look at the rate, it's still around 13.7 per 100,000 people. So why is that, even though this number went down? It turns out the diagnosis rate is the number of new diagnoses, new cases, over the whole population, times 100,000. But here, even though the number of new cases went down, the population has grown significantly, so that is why in the end we still get about the same rate. So population growth can cause incident rates to stay steady, even if the absolute number of new cases goes down. And for chronic diseases, prevalence is always greater or higher than incidence because the number of cases can accumulate or add up over time. This figure shows HIV incidence, which is indicated in blue, so this is incidence, versus prevalence, which is indicated in the red line. And I can see the number of cases is shown in the y-axis and the years in the x-axis. What you'll notice is that prevalence steadily rises because people live long-term with HIV, Incidence fluctuates from year to year, reflecting new transmissions. And for a chronic disease like HIV infection, prevalence will generally be higher than incidence because it represents the cumulative number of new cases over many years, minus the number of cases that are no longer active because the patient either died or was cured. Mortality is the number of deaths due to a disease, and the mortality rate is again usually expressed per population size, usually per 100,000 people. Comparing morbidity and mortality helps to determine the disease severity and the adequacy of prevention or treatment. This first checkpoint asks, explain the difference between incidence and prevalence. So incidence is the number or proportion of new cases or disease that appear in a population over a specific period of time. Whereas prevalence is the number or proportion of existing or total cases of a disease in a population at a given point in time. And because prevalence includes all of the incidents or new cases, plus any older cases that are still active, then it must always be greater than or at least equal to incidence. For chronic diseases, as I mentioned, like HIV, prevalence is higher than incidence because cases will accumulate over many years. For the second checkpoint, they ask, describe how morbidity and mortality rates are expressed. So morbidity rate is the number of diseased individuals in a population, and it's expressed as cases per 100,000 people or as a percentage of the population. Mortality rate is the number of deaths caused by a disease, also expressed as deaths per 100,000 people or a percent of the population who died from the disease. And these standardized rates really offer or allow comparisons across different populations and over different time periods. Now let's look at patterns of incidence. This is the occurrence or rate or frequency of a disease, and there are 4 classic patterns. Sporadic diseases are rare, and there is no predictable pattern because it's irregular, no geographic pattern as well, and examples include tetanus, rabies, or plague that you see pop up here and there in the news. Endemic diseases are consistently present at a stable level. So this is predictable, it's occurring at the rate that epidemiologists predicted they would occur. And an example is malaria in parts of Brazil. In epidemics, we see a sudden increase above expected levels. And an example could be seasonal influenza outbreaks that are unexpected. And then pandemics are epidemics that spread across continents. So this is occurring worldwide at different continents, and these include cases like HIV or AIDS, as well as COVID-19. And understanding these patterns really help health agencies anticipate the resource needs. This graph shows an example epidemic which occurred during that 2007-2008 US influenza season. We can see that emergency department visits jumped above normal levels due to circulating influenza strains that partially escaped immunity. And this illustrates how viral evolution, remember that antigenic shift and antigenic drift, can break the usual disease equilibrium. In that season, what happened was the vaccine match against the virus that was out there that season, H3N2, wasn't quite optimal. There was also an increased resistance in the H1N1 strain to the current antiviral meds that were available during that time. This graph shows a percentage of all deaths due to influenza and pneumonia, and the CDC sets an epidemic threshold. This is what they expect to see based on the previous 5 years of data they have. And when mortality exceeds this threshold, the situation is considered an epidemic. So we can see a few cases where the mortality line, exceeds the expected value, that expected range, and those are considered epidemics. So there are clear epidemics in the 2003-2004 season, in 2005, as well as 2008. In most years, diseases will exist at a stable or predictable level, and those are endemics. So this is again when the disease stays at a steady, expected rate in the population. That is endemic equilibrium. For example, influenza season happens every year, and it's usually at a predictable level because we have some immunity from past infections or from the vaccines we get, and the virus has not changed dramatically. So the environment and the population behavior are relatively constant in this situation. But an epidemic occurs when that equilibrium is broken and cases rise much higher than expected. So what this tells us is the normal balance between the pathogen and population immunity has been disturbed. And this means something changed. Because of this change, the pathogen can now infect many more people than usual. And this can be, for instance, due to those mutations or resulting from antigenic drift or shift. Now the virus can hide from or evade from our immune memory from previous seasons or vaccines, and that's when we get those epidemics. So we saw that epidemics occur when the usual balance between the pathogen and host immunity is disrupted. And for influenza, antigenic drift and shift can help the virus evade immune memory, causing seasonal epidemics and occasionally even global pandemics. Again, this is when we see this spread across continents. And as we saw in the previous graphs, CDC thresholds define when seasonal incidence crosses epidemic levels. All right, this next checkpoint asks, explain the difference between sporadic and endemic disease. So sporadic diseases occur only occasionally and without geographic concentration. Things like tetanus, rabies, or plague that pop up randomly with no pattern. Whereas endemic diseases are constantly present, often at a low steady level within a particular region, such as we're seeing malaria in parts of Brazil regularly. For the second checkpoint, they ask, explain the difference between endemic and epidemic disease. So again, endemic is a baseline steady pattern of disease presence in an area that you expect. And this expectation is based on previous years. Whereas an epidemic is a sudden increase, a larger than expected number of cases in a specific area and in a short time period. For example, we just saw that seasonal influenza can become epidemic in winters when the case numbers rise well above what we expected. Etiology means defining the specific pathogen or etiologic agent that's causing the disease. And Koch's postulates, as we saw previously in Chapter 15, historically linked microbes to specific diseases, but the modern molecular versions rely on evidence at the level of genes or traits. But there are challenges, and these include overlapping symptoms, slow-growing organisms or organisms that can't be cultured, and the need for laboratory confirmation. This next checkpoint asks us to list some challenges to determining the causative agent of a disease outbreak. So they're asking us to think about why etiology or determining the cause of disease can be difficult. So one is nonspecific signs and symptoms. Many pathogens produce similar signs and symptoms, and that can complicate diagnosis. Second one is there could be multiple possible causes, and we have to remember that association is not the same as causation. We often need to perform additional controlled experiments. A classic example is the association observed between ice cream and polio. So it turns out that in summer months, the number of polio cases increased at the same time that ice cream sales increased. And this is not causation because ice cream does not cause polio. Polio is caused by poliovirus. Both were simply related to warm weather. In warm weather, we saw more people outside. That led to more person-to-person transmission. And poliovirus happens to be stable and survives well in warm, moist environments. And then there was also more ice cream purchases because it was hot. So the takeaway there was a shared seasonal pattern that created a false association. For the third one, there could be detection difficulty. Some pathogens are hard to isolate or hard to identify early in an outbreak. And then sometimes, or most of the time, we also need laboratory confirmation. So epidemiological data alone cannot prove causation. So outbreak investigations always rely on a combination of clinical interviews, lab tests, and pattern recognition. The CDC, or the Centers for Disease Control and Prevention, is the primary U.S. public health agency, and they lead the National Disease Prevention and Outbreak Response. They also oversee the National Notifiable Disease Surveillance System, or NNDSS, and clinicians and labs have to report cases to local health departments. States then report aggregated data to CDC via this NNDSS, this National Notifiable Disease Surveillance System. And then the CDC will share data with the World Health Organization for global surveillance. So if you're interested, you can go to this link over here to learn more about the NNDSS data, and you can also see the list of notifiable diseases at the link here, and you can search the list by year. So I went to the link and searched for the notifiable or reportable disease lists for the year 2025, and it was very long. This is a screenshot of a few sections of the long list shown here. And this is a screenshot of the National Notifiable Disease Surveillance System annual summary data. You can also search here by disease, year, and other filters. And I searched for all diseases, and this is an annual summary for 2022. You can see the number of different cases of diseases for this year. Here I scroll down. This is the same summary from 2022, looking at different diseases. Total botulism, we had about 197 that year. Candida, this yeast infection, we had about over 1,000. And the total versus confirmed cases of COVID, you can see, are in the tens of millions. The CDC also publishes weekly this Morbidity and Mortality Weekly Report. It's the CDC's major surveillance publication and provides more rapid updates so clinicians, health departments, and hospitals can respond to emerging trends. And if you're interested, I provided the link here to explore further. And this table from our book shows incidence trends from January 2016. So this is one part of that Morbidity and Mortality Weekly Report, or MMWR, from that time. And notice the scale for the diseases. Some diseases like chlamydia have very high incidence compared to others. So chlamydia, we had more than 1.4 million cases, gonorrhea almost 370,000, giardiasis almost 12,000. So this report really helps public health workers to track abnormal increases or spikes in a disease. This checkpoint asks, describe how health agencies obtain data about the incidence of diseases of public health importance. So the Centers for Disease Control and Prevention, or the CDC, oversees that National Notifiable Disease Surveillance System, the NNDSS. Physicians and labs are legally required to report cases of those notifiable or reportable diseases, such as HIV or measles, and we saw that long list. Then the CDC compiles and publishes this information in that Morbidity and Mortality Weekly Report, that MMWR. And these reports help public health officials track trends, identify outbreaks, and guide prevention measures. In this chapter, the book also takes us through a classic foodborne outbreak investigation as the clinical case study problem. There was a local spike in patients coming in with gastroenteritis-like symptoms like vomiting, diarrhea, stomach cramps, severe dehydration due to that diarrhea and vomiting. And then they did some lab testing of the stool samples from these patients and found salmonella bacteria. Then epidemiologists selected a random sample of those patients to interview, and they found out that all of those patients ate turducken. So I Googled and put a picture in here. Turducken is a chicken stuffed inside a duck, and that is stuffed inside a turkey. Then the epidemiologists found the source of contaminated turduckens. It was in a facility where their machine was contaminated with salmonella bacteria. They also had substandard cleaning and production protocols. So these epidemiologists were able to link that gastrointestinal illness to contaminated turducken through those patient interviews and then also reviewing their health records and lab tests. So this illustrates the combination of pattern recognition and microbiological confirmation that really defines epidemiology. All right, let's recap 16.1. The first learning objective asks us to explain the difference between prevalence and incidence of disease. So prevalence was the total number or proportion of existing cases, including both old and new cases of a disease in a population at a given point in time, whereas the incidence was the number of new cases of a disease that develop over a specific time period. So the key distinction is prevalence measures how widespread a disease is, and incidence measures how fast new cases occur. For the second learning objective, I'm going to skip this for now because I have that table to show you, so we'll do that in the next slide. So let's look at the third one. Explain the use of Koch's postulates and their modifications to determine the etiology of disease. So Koch's postulates were a set of 4 criteria that we use to prove that a specific microorganism causes a particular disease. And we saw this again in chapter 15. So this included 1, the suspected pathogen must be present in all cases of the disease, but absent in healthy individuals. Then we had to isolate and grow the pathogen in pure culture. Then we had to take that cultured pathogen and make sure it causes the same disease when introduced into a healthy host. And then the same pathogen has to be re-isolated from that newly infected host. For the more modern version, that was molecular Koch's postulates, and those used genetic evidence to link specific virulence factors or genes to a disease when traditional culturing or infection steps were not possible. So again, these postulates help us identify the etiological agent or etiologic agent, that causative agent, the pathogen causing the disease or outbreak, and helps us confirm direct causation rather than correlation. Finally, our last one, explain the relationship between epidemiology and public health. So epidemiology studies the occurrence, distribution, and transmission of diseases within populations. And findings from epidemiological research inform public health decisions, such as identifying outbreaks, tracing sources, and planning prevention strategies. We have our public health organizations like the CDC, the World Health Organization, who rely on this data to monitor notifiable diseases and publish reports like the MMWR, and also develop control measures. So in short, epidemiology provides the scientific evidence, and public health applies it to protect communities from disease and injury. And stepping back to that second learning objective, distinguish the characteristics of sporadic, endemic, epidemic, and pandemic diseases, I took the table we saw previously in this video and blanked out the description and examples to see if you can fill them in. Remember, our sporadic diseases are those that occur rarely and irregularly. There was no geographic pattern like tetanus or plague. Endemic was constantly present in a region at a predictable low level, like malaria in parts of Brazil. Epidemics, you see a sudden increase in the number of cases way above expected levels in a specific region and time period, like our seasonal influenza outbreaks. And pandemic, these are worldwide epidemics that affect multiple continents, like COVID-19. So see if you can try on your own and define each of these and provide examples for each as well. And that concludes section 16.1. In the next video, we'll move into 16.2, which is tracking infectious diseases, where we apply these concepts to real surveillance systems.